Hello, good morning, students. Hi, how are you? Okay, good morning. Okay, I hope that today you're all ready for today's class, and I hope you've also revised what we have done for the first lesson, which is we have started chapter 11. So today is a continuation of the previous lesson, which was on last Saturday. Good morning, girls. Right. Good morning, Ke. Good morning, Ninin, and everyone else who are here, who are watching. Okay, so today we are going to learn how to draw a diagram to show a kind of inheritance we call monohybrid inheritance. Good morning, Jen Shuan, Jiawei, Xin Hui, Angela, Yumin. Okay, everybody, good morning. Okay, now I hope you're all ready for class. And the first lesson, what I did was I explained all the meanings of the terms that you're going to use today. Okay, so you have to really understand the meaning of the terms before we start today's lesson, because otherwise you'll be lost. Okay, Macy, good morning. Right, let's start the lesson. I hope you can see this. I'm going to share screen now. Okay, so today we're going to learn how to draw this diagram to show monohybrid inheritance. Okay, don't worry, I'm going to explain to you what it means by monohybrid. Okay, just in a while. All right, now, before that, I hope you've already understood all these terms, which I already explained in my previous lesson. If you have not understood, okay, it's good that you have not seen it. Just go back to see and understand the terms first, right? Because I'm going to use all of it, most of it in today's lesson. And previously, all right, let's revise a little bit. Previously, about when we want to talk about characteristic, okay, we have traits. Traits are variation of the characteristic. For example, we talk about seed shape. Okay, so Mendel is the father of modern genetics. So he did a lot of investigations on this Pisum sativum. This is kacang. We call it green. We call it green peas. Not really green peas. We call it snow pea. We call it. The actual name is called snow pea. So what he did was he took different characteristics of this, for example, let's say smooth seed and also wrinkled seed, and then he crossbreed them. He crossbreed them, and then he wanted to find out how many percent of the children, or not children, say offspring, offspring of the means the next generation will show the smooth trait, and how many will show wrinkled trait. Okay, so he did a lot of investigation. So these are the 7 types of characters or characteristics that he investigated with this kacang, right? We call it Pisum sativum. So it's seed shape. There are 2 types. So they are called traits. So smooth and wrinkled. Okay. For seed color, there's either yellow or green. Then the pod shape, this is actually the fruit, okay, where the seeds are inside there. It's either inflated or constricted. Inflated means it's bloated. It's like, kembang like that, right? It's like a balloon, right? And in constricted, it's all flat, and it's like, it's already sort of wrinkled. Okay, so pod color is green or yellow, and purple or white for the flower color. And flower position is axial or terminal. Terminal means the flowers at the end. Axial is like in between the branches. Okay, the flower can also grow between the, that there's flower in between the branches there. And the height is either tall or dwarf. Okay, dwarf means short. Okay, let me see what's okay. Good morning. Right now, so when you learn about these traits, we learn that there were alleles. Alleles are these genes, alternative forms of the gene. So we've learned there's dominant and recessive. Okay, so Mendel found that not all the genes or not all these factors are equal strength. There are some which are stronger, we call it dominant trait, and there are some which are not so strong, we call it recessive trait. Okay, so you have this dominant and you have this recessive. So usually you use a capital letter to show dominant trait, dominant allele, sorry. So let's say, for example, capital B. Okay, capital B for dominant, and a small one for what we call recessive allele. So when it is yellow, it could be either one of the 2. You have that 2 also capital letter, or 1 big and 1 small. Okay, because dominant is stronger. So if you have 2 copies of the dominant allele, it will definitely show the dominant trait, which is yellow. Then if you only have 1 that is dominant allele, 1 recessive, it will still show yellow, because yellow is known to be dominant. Okay, so even if you have only 1, which means capital B, all right, doesn't matter what is the other one, whether it's a recessive dominant, as long as you have one dominant allele, it will show the trait, which is yellow. Okay, so for all these dominant traits, you have either both the same, let's say capital R, or you can also have one capital R and small r. So these are two different types of what you call the genes that you have for dominant trait. Okay, whereas for recessive, there's only one choice. You need to have both recessive trait alleles to show the recessive trait. Okay, so you need to have both copies, because if you have one of this, it shows the inflated. It already shows this one, which is because this is stronger. Okay, so make sure you know this very well, because we're going to look at this afterwards. All right, if you haven't, like I mentioned, after this, you please go and revise that. Okay, now today we're going to learn about monohybrid inheritance. So what is monohybrid inheritance and how does the diagram look like? So I'm going to show you how to draw this kind of diagrams. Okay, the diagram is like this. In your textbook, like this. Okay, you need to show what is the type of, what are the types of offspring these parents can produce. Okay, so we're going to have, this one, you're looking at two generations. This is the parental means the parents. Okay, this is the parents here. So the parents here, you have purple and white. You cross them, you will get what we call F1. F1 is called first filial generation. That means this is the children, the first generation. Okay, simply said, it's just like the first generation. So you're going to get all purple flowers. Okay, so he did the investigation many times, and then he realized that in the first generation, Mendel found that he only have one color only. In the first generation, there's no white. All of them happen to be purple. Okay, so he repeated many times, and he found that this is always true. Then after that, he used the purple flowers, all right, and he self-pollinate. That means self-cross. Means take the pollen grain from the flower and put it back onto the stigma of the same flower. That is called self-pollination. And he found he's going to get the next generation. Okay, the next generation is called F2. Okay, F2 is called second filial generation. So the second filial generation, we have a combination. He will not get all purple, not get all white, but rather he found that there is a ratio, and the ratio is always 3:1, where the 3, this is the more dominant. The purple, he said, is more dominant because it like happens to have more of it. Okay, so he found that that means this allele for the two types of colors, they are not equal strength. There will be one which is stronger and one which is weaker. So he said that the one that appears to be 3 must be the dominant one, and the one that is 1:1 must be the recessive one. Okay, so this is monohybrid inheritance. So today we're going to learn how to draw this properly and how to label it with all this at the side. This you have to, otherwise you do not get your marks. It's not enough for you to just give me this. It is not enough. You have to write this one every level. This line represents what? This is gamete. This line represents fertilization. This is phenotype and genotype. This is phenotype. Okay, don't worry if you don't know what I'm talking about. I'm going to teach you how to do it. Okay, so let's look at Mendel's monohybrid inheritance. Okay, it's his experiment. So let's look at this. Now, Gregor Mendel, why did he choose to experiment on Pisum sativum? Why he didn't use mice, or he didn't use animals or any other organism? Why did he choose Pisum sativum for a subject of investigation? Okay, to find out the percentage of how many, the parent like this, the father's like this, the mother's like this. Okay, what happens to the children? Okay, why didn't he use animals? Of course, it's more interesting to use animals, but it's also more difficult to handle. Okay, using animals is more difficult to handle. Okay, so for plants, number one, of course, they don't move, right? You don't move, you need to have to chase them around. All right, it's easy to grow. Okay, they grow quite fast as well. And the life cycle, this is important. Life cycle is short. That means in a span of about 2 months, 60 days is about 2 months. You can start. You can have, after 60 days, you can have the next generation already. That means when you start investigation, you start to breed your plants, right? Let's say you take the pollen grain from this anther, you put it onto the ovary. That's where you start to cross. Okay, you take let's say this is what he did. He took the anther of the flower that is already matured. He took a brush. He brushed it onto the stigma. Sorry, that's not the stigma. Yeah, he brushed it onto the stigma. So this is the stigma of the female. Okay, first he did cross. That means different flower. Okay, different plant. Take the anther, the pollen grain from this anther, this flower, the anther, right? Then he took the pollen grain and then he put it onto the stigma of another plant, same species. Okay, we're talking about same species, different plant, different flower. So that what he did is to cross pollinate. Okay, so he did it manually. And after that, of course, this ovary, after the pollen grain came, right, it will be fertilized by the pollen grain. So this ovary will develop to become the fruit. So what is the fruit of this pea plant? The fruit is the one that we eat. This is the one, this is called the pod. You eat the we call it the snow pea, isn't it? This one. Inside there got the green color peas. Yes, the whole thing is green, right? If it's the green variant. So this is the whole fruit actually. So we call it the pod, P-O-D. All right. So already got the seed. This is the seed. This is the seed, right? The seed is inside the pot. This is the seed. So now what he did was, of course, you don't harvest them, you don't cut them, and you don't plant them, don't eat them, right? What you do is wait for them to become old. Wait for them to become old, mature. Right? I mean, not to say mature, it become old. Then you take out the seed and you plant them. So this is the anak. Okay, this is the what you call F1 already. Why you plant them? Because if you don't plant them, how you're going to know whether this offspring is going to have purple flower or white flower? You must give it a chance for it to grow. Okay, so this is a seed. This is the F1 already. You must let it grow. Okay, let's say now it grow into a plant. Okay, grow into a plant already. Then you can see the flower. Then you see how many percentage of it is purple flower. How many will have white flower? That's how it is. That's how he did the experiment. I'm just explaining to you how he did the experiment. Okay, now he didn't stop there. This is your F1. He carried on the experiment until F2. That means the next generation, the two generation. Okay, the grandchildren. So what he did was now he took the F1, right, whatever plant there is, whatever plant, he did a self-pollination. That means pollinate same flower, take the pollen grain of the same flower, put it back onto the stigma of the same flower, okay, or maybe different flower but the same plant, all right, because self-pollinate. And then after that, the flower will, the ovary will be fertilized. You're going to get another generation, so you're going to get a pod. Okay, we'll get some pods. This is the pod, right? This is the pod. So this is going to be F2. Okay, so you wait for it to, fertilize and all that, everything. You're going to get your pot. Now, after that, same thing repeated. He took out the seeds and then he planted. So when he took out the seeds and then he planted them, all right, this is the plant. All right, let me just show the plant. So this is going to be your F2. So he planted all of the seeds and see how many percentage of this F2 will have purple flowers, how many percentage will have white flowers. Okay, so essentially this is what he did. Okay, he first of all, he crossbreed two parents of different traits, one purple flower, one purposely he choose different color so that he knows what happened to the children. So one is purple, one is white. Okay, father purple, mother is white, let's say. Okay, so he took the pollen grain from the father, from the male, then he put it onto the stigma of the female plant, the flower of the female plant, fertilize, then you get F1. He took out the F1, he planted it, because if you don't plant it, you can't see from the seed whether it carries the purple gene or the white gene. You don't know, isn't it? You have to take it out and you plant it and let it grow. And the good thing is because they grow very fast, so it doesn't have to wait very long. So your investigation can be finished in, let's say, 60 days, you can have one generation already. Another 60 days, you can have the next generation. So you can complete the experiment very fast. If he had used animals, or he used humans, even teruk, even worse. Because humans, you see, you take about 20 years for a baby to grow, become adult. Then only you can the adult can have the next generation, isn't it? You got to wait 20 years. Now, who has the time to wait for that? You need to do repeated investigation, repeated trials. So you can't, if your experiment takes such a long time, you cannot have enough data for you to come up to a conclusion, isn't it, right? So you see, using plants is simple, and you must use a plant that has a very short cycle. That means it can complete one generation within a minimum amount of time. Okay, so he chose Pisum sativum, but that's not the only reason. He's got other reason why he chose Pisum sativum. Okay, I'll go to that. So this is essentially what he did, right? He took the seed and then he plant, and then he got the next generation, and then he counts and see how many with purple flower, how many with white flowers, or how many are tall, how many are short. Okay, so that's one reason, short cycle. Life cycle is very short. Number two, the pollination can be controlled easily. Like I mentioned, you just take, if you do not want self-pollination, you just cut off the anther from the same flower, because you want to do cross-pollination. So he doesn't want it to be the first generation that you want to get F1, you have to do cross-pollination. You cut off the stamen of the plant, and you do not want its own pollen grain to drop onto the stigma. So you just snip off the anther. It's easy to control what kind of pollination you want. Okay, if you want self-pollination, you don't cut off the anther of the you just, you can put it back onto the stigma. If you want cross-pollination, you make sure you don't want it self, you just snip off your filament. Okay, next one, D have both male and female parts. And number E is very important, are the traits that he wants to investigate. They are contrasting. Contrasting means this: the purple and white flowers, isn't it? For pea plant, there's either purple or there's other white. There are no flowers which are in between or has a combination of white and purple at the same flower. So it's either this or that. These are really contrasting. And for not only flower color, there are many contrasting traits. Okay, like I mentioned here, this is the contrasting traits. Flower, you have either purple or white. And then for the pot color, it's either green or yellow. There's nothing in between. So it's easy to see, easy to investigate. Inflated or constricted for the shape of the pot. Okay, and then the seed color, yellow or green. Then for the flower position, whether it's axial or terminal, it's either this or that. If it's not this, it must be that one. So it's very easy to see or identify your characteristics. And height, either they are tall or short. Okay, or dwarf. That means there are two types of Pisum sativum plant, whether it's tall or whether it's short. There's nothing in between. There's no medium. Okay, it's either tall or it's either short. So that's why he chose Pisum sativum, all right, Mendel, to do this investigation. Okay, so now let's talk about, let's revise what is self-pollination and cross-pollination. Okay, now let's say I have two plants here, all right? You learned it in form 3 actually. Okay, no, form 2. Sorry, form 1. Your new syllabus is in form 1. The old syllabus was in form 3. Now, what is self-pollination? Okay, let's look at, you have two plants here, right? Two different plants, but same species. Okay, make sure you crossbreed the same species, because if you're doing something different, it's not within this inheritance already. We're talking of the same species. So you have a plant, okay, a plant A, and plant B. All right, plant A, plant B, same species. Now this happens to be bougainvillea, okay, but doesn't matter lah. This is only suitable picture I found. Now let's say you have many flowers on one plant, isn't it? One plant doesn't have only one flower, so you have many flowers. Let's say flower X, flower Y. All right, I just took 2 flowers from there. Now, if it's considered a self-pollination, there are 2 ways to have self-pollination. Number 1 is the pollen grain of the anther. Let's say I'm drawing a flower here. Okay, I'm drawing another X, this is Y. If it's in order for it to be self-pollination, it can be like this. Number 1, first method is you take the pollen grain of flower X, it drops back onto the stigma of the same flower. This is if the flower is bisexual. Bisexual means it has got male and female parts. Okay, so it drops back the flower, only one flower. All right. So the pollen grain from the flower drop back to the same the stigma of the same flower. This is called self-pollination. Okay, this is quite clear lah. Of course, self, isn't it? Now it can also be another way where the pollen grain of plant X, the flower X, it went onto another flower, but still on the same plant. This is still considered as self-pollination. As long as on the same plant, doesn't matter you have different flower. because they will carry the same gene. Okay, different flower, but it will have the same gene, all right, because it's the same plant. So this is also considered self. Now, how is it considered cross? A cross means must be between different plants. So let's say you take the the pollen grain of this flower, and you put it onto the stigma of another plant. So this is considered cross. Okay, so I want you to set the picture clear so that you know what is it considered self and what is considered as cross-pollination. All right, so cross-pollination, make sure it is of a different plant. Same species, make sure it's same species, but different plant. Okay, it has to be on a different plant. That is considered cross-pollination. So first of all, let's see how Mendel did the experiment. Now, so here, sorry, I have to make it bigger. So cross-pollination is the process when one plant, you see different plant, fertilizes another plant, other plant. Same species, this is for same species, but cross-pollination, it can carry on to different meaning. If we're talking about crossbreeding, that's crossbreeding. Crossbreeding means different species. Now this is same species, different plant, okay? Self-pollination is a process where the plant fertilizes itself without the involvement of other plants. So that means same plant. Okay, so Mendel chose pure breeding, pea plant. When he started his experiment, he chose plants which are pure breeding. What is the meaning of pure breeding? Pure breeding in Mandarin we say something like 它的种是纯的咯，纯 means pure. That means it didn't have any other genes, okay, to show there's a combination, it's not a mix. In other words, to say simply, it's not a mix. It has pure genes. Okay, for example, what do you mean by that? A pure breeding plant is obtained after many generations of self-pollination. How do you know whether that plant is a pure breed? Okay, very simple. Let's say, I give you this scenario. If I want to know whether this plant is a pure breed or not, what I do is I take the pollen grain of this flower on the plant, and then I pollinate I put it on the stigma. So that means self-pollination. Okay, self-pollination. So self-pollination, and then I take the next generation and I plant it. Okay, self-pollination. And let's say the next generation, we call the F1, has the same characteristic as the parent. T is a parental. So okay, it looks the same. Let's say tall. I take a tall plant, I self-pollinate, and I found that the children will have all tall. Then again, I repeat, and then I self-pollinate the F1, and then I'm going to get my F2, isn't it? The next generation is called F2 lah. And then I find, let's say, is also tall. All of them tall. None of them exhibit the short, we call it the short traits. None of them. Okay, let's say I'm still not satisfied. I want to be doubly sure. Okay, so I do again another time. For the F2, I crossbreed. Crossbreed. So I self-pollinate. I'm going to get my F3, right? The next generation is the F3. Let's say I also get them tall. None of them are short. What is the conclusion I can come to? What is my conclusion? My conclusion here is, if I do it many, many rounds, okay, I crossbreed, I want to see what is the kind of gene that can come up from that. I mean, what kind of traits I crossbreed many, many times. I always get tall and tall and tall and tall. So what is the conclusion? The conclusion is this plant must be only having tall genes. That means it didn't have any short or dwarf genes inside this plant. It only carries tall genes. So if it only carries tall genes, for example, I can conclude that the allele must be both also tall allele. It is not of this type. Not this type. Because this one is a recessive. This is, let's say, dwarf. Okay, tall is, Mendel has found that tall is dominant, and this is dwarf, let's say. If it is this type, definitely, if it's this type, okay, if you started with this one, if you started with this as a parent, the next generation, maybe you do not get short, but the next generation, you may have short already. It will appear. Because if you have that gene, somehow or other, along the generations, this gene will show itself. You will show the shortness there. Okay, so after self-pollinating for so many rounds, he found that it doesn't appear to have any short features. So that means this plant, the plant that you started with, must be a what you call a pure breed. So this must be pure breed because that means it only has one type of allele, which is the tall allele. Okay, so he can come to that conclusion. So you want to know before you start investigation, he started with pure breed first. So he must make sure that that plant is pure breed. So before that, he must do some prior investigation. He crossed self-breed, self-pollination, many rounds, many generations. He found that this one can only produce tall. That means this one must be a pure breed. Okay, so what he did was he took the pure breed tall. Yeah, you look at this. This is from the textbook. So the tall feature. This is a pure breed plant A, let's say, and then he cross breed with a short one. He took a dwarf. So this one also he tested first. He must test that it is dwarf first. Many generations also dwarf. All the plants will come out dwarf. That means it must be a pure breed. Okay, pure breed dwarf. He purposely wanted to see. He was very curious. Okay, so Mendel is a very interested man who's very interested in maths and science. In fact, not just science, and he also interested in maths and meteorology. He did a lot of investigation, and he was actually a very smart person. Can do that. Yeah, he came up with his Mendel's first law and second law. Okay, after doing thousands of investigations, thousands of in these cross breedings. Okay, so tall and dwarf. Now this is called a monohybrid inheritance because we are only looking at one feature. We're only looking at whether it's tall or short. We're not looking at the flower position at the same time. We don't care where the flower position is. In our investigation, we only want to investigate whether what is the inheritance for tall and short. Okay, so this is called mono, one feature only or one characteristic we are investigating. So monohybrid inheritance refers to inheritance of one character. We are only concerned about how what is the ratio of this tall or short in the next generation. We don't care whether the purple flower or white flower. We don't want to care to see whether it is got green pea, the pea pot color is green or yellow, or whether the shape of the pot is round or smooth or wrinkled. We don't care. We just want to see one thing only at one time. Okay, so this is called mono. Mono means one. Okay, so we are only learning at monohybrid inheritance. Next week we do dihybrid because there'll be two characters we are investigating. Okay, so monohybrid inheritance, what does it mean? It's a genetic cross between two individuals that involves the inheritance of one characteristic only. So we're looking at one character, which is whether tall or short. No need to see other things. Okay, so he chose the height. So first of all, let's say he's doing one of his very famous investigations. He did height. Okay, he purposely chose pure breeds of the tall, and then he crossbreed with the short plant. Okay, so he cross-pollinated the tall and the short. The next generation that is produced are called first filial generation, or we say F1. The next one that came out. Okay, after that, from the F1, okay, he took out all the peas from the F1 and then he planted them and then let them grow. After that, he took the what they call the pollen grain of the plant and put back onto the stigma of the same plant. So he did a self-pollination for the F1. F1 he self-pollinate. Parental he's cross-pollinate. Okay. Then after that, got the seed. Then he planted. And then he got his second generation called the second filial generation, F2. Okay, the next generation is called F2. And then he noticed something very peculiar. He noticed something very weird. He found that no matter how many times he repeated the experiment, he will always get the second generation, the ratio of these characteristics. It will not be all tall, it will not be all short, but he will always get 3:1. And that intrigued him. He didn't know why was it always that. If he is always it always happens that way, there must be a law. There must be a law to it. So he devised from that, he deduced his first law of inheritance. Okay, and then later I'm going to discuss with you what is Mendel's first law of inheritance. So 3:1. Okay, first of all, let me teach you how to draw the diagram. Okay, so let's do it this way. First of all, you must write your key. Key is a petunjuk. What does your symbol stand for? Your capital stand for what? And your small t stand for what? So since we're using tall, let's write T lah, okay? And definitely T, there's no mistake about capital and small. So I suggest that you use your capital T and small. Don't use other symbols like I mentioned here earlier on in my lesson last week, last lesson. There are certain symbols that you don't use. For example, your C and your C. Because sometimes, you know, the way you write your C, I do not know whether it's big or small. It could look like big, it could look like small, and you're going to confuse me, and you're going to make yourself risky, all right? Or you put yourself at risk of getting it wrong, because the examiner can misinterpret your symbols. So don't use all the others that are crossed out. Use all the ones that I've highlighted, because they are very different, like H and H. You can, no mistake about it. Okay, now let's look T, tall. So Mendel said that perhaps one is more dominant, so we put a capital letter to represent that, capital T. Capital T is a dominant allele for tall, and small t is a recessive allele for short. Okay, now, so he found that this is the only way to explain why you get 3 to 1. In fact, it's very smart. If you had not come up with this thing, you cannot explain why you will always get 3 to 1. So he came up with his law. Okay, now the law is Mendel's first law. Let me go to the law first before I go to this diagram, because you need to know the law before you draw this diagram. Okay, let's look at the first law. Mendel has his first law of inheritance. Okay, it's called first law of inheritance, or we call it law of segregation. He said that, okay, what is the thing I need to underline this, I need to remember. He said that a characteristic inside an organism, okay, a characteristic, whether it's tall or short or whatever, all right, purple flower or white flower, it is determined by a pair of alleles. There is a pair, okay, not just one. For example, look at this. This is an individual that is tall. Okay, let's say tall. You see you have two inside there. There is a capital T and a small t. Now why? This is because of homologous chromosome. Okay, remember we in order to form a child, you need to have a father and a mother, correct? The father will supply the gene through the sperm. Let's say talking about animals. Sperm carries the father's genes. And the mother will carry, of course, the ovum will carry the mother's genes. So you need to have the gene that's carried by the sperm, right? And also it will fertilize the gene that's already in the ovum. So that's why the father, this one came from the father, let's say, and this one came from the mother. You must have a copy of the gene from the father, which is called the maternal, all right, for paternal, and then the mother is maternal. So you must have 2 sets of it. All right, no, sorry, one set means 2 copies. So that's why in the alleles, you must also have 2 alleles. One allele you got from the father, okay, let's say the capital T. And then the other allele came from the mother. Okay, in terms of plant, we call it male and female. You're talking about humans, animals, say father and mother. So you will have 2 copies of allele. Okay, so that's why this is the first the statement that you need to know. Any organism, the characteristic that is determined, the alleles that are the genes that determine the characteristic, they must be 2 copies of it. That means there are 2 alleles. And in the formation of gamete, let's say you want to make the next generation, you got to form gamete first, isn't it? Right? Remember, in the formation of gametes, you must have meiosis. Remember, you must have meiosis. Otherwise, you cannot form your gametes. You must have meiosis. And in the process of meiosis, right, the allele will now separate. Okay? Just now you have capital T and small t. So, in order to form gamete, the gamete you learn in your Form 4 work, you remember when the chromosome split, all right, the chromosome split, the homologous chromosome split during metaphase 1, one will go to one cell, one will go to the one end of the cell and go to the other end. In the end, you're going to have 2, isn't it? So you're going to have 2 gametes. Now, in those 2 gametes, the gamete can only carry 1. Okay, this will carry, let's say, the capital G, the capital T. This one carries the capital T, and the other gamete will carry the small, the recessive gene. So you see, in the formation of gametes, the pair of alleles will segregate. It will segregate. It means separate. Segregate is the same meaning as separate. It has to separate. There is no such gamete that carries 2 together. Because if we carry two together, that means it's a chat one, right? Gamete must only carry half the number of chromosomes. If we carry two, that means it's a full set. It is 46 already. It's not 23 chromosomes. Okay, so it only carries half of it. That means the allele must separate during meiosis 1, and only one allele. There's only one allele in the gamete. One gamete can only carry one allele. Okay, so by understanding this, now we can go and draw the monohybrid inheritance diagram. Okay, so remember, it must separate during meiosis, and in the formation of gamete, only one gamete can carry one allele. It cannot have capital T, small t. It has only one. Okay, now let's look at this. Let's look at how to draw it. So first of all, you write your key. Okay, your key is already there. So, oops, sorry, it just jumped. Okay, so your key is you must state what your symbols represent. So your dominant, capital T is dominant allele, definitely dominant when it's capital, and then small is recessive. Okay, so how do you get 3 to 1? The only way to get 3 to 1 is by this explanation. Okay, first, he started with pure breed, isn't it? He started with pure breed, that means this parent must be only carrying 2 copies of the same type, tall only. It only has got tall. So we call this homozygous. Okay, you learned that before. This is the last week's lesson. Sorry, homozygous means both the alleles are the same type. And for the pure breed that is short or dwarf, he also used pure breed. So it must also be small t, because this is pure breed. There's no mix of the tall gene there or the allele there. So this is also homozygous. Okay, it's homozygous. Where is it? Yeah, homozygous. Okay, now next, in the formation of gamete. Now this one doesn't show. I'm going to draw separately. It doesn't show how you get this. Okay, I'm going to draw it here. All right, I'm going to draw it here easily. Okay, so you have your parent, capital T for one of the parent. You cross with a small t, both are pure breeds. Pure breeds. Okay, pure breed. This is also pure breed. So this is parent. Okay, it helps if you were to draw as I'm going along. It sort of like makes you understand a bit better in this to draw it. Okay, so this is the parent. This is a parent. In order to produce the next generation, you must have meiosis to produce the gamete. So here you're going to draw 2 lines to separate, to show the formation of gamete. So this line represents meiosis. The 2 arrows there, in fact, the 4 arrows here representing meiosis. Okay, now then you're going to get your gamete. So according to Mendel's first law, it states that in any gamete, it can only carry one allele. So it has to separate. So the big T goes here, the big T goes here, small t goes here, small t goes here. So this is going to be the gamete. So write down at the side here. This is the gamete. Okay. Now, in order to form the zygote, okay, of course the gamete has to combine with the male gamete has to combine with the female gamete. So let's say this is the male plant. This is the male plant. This is the female plant. Okay, so you got to combine. Now, what gametes can combine with each other? You need to combine them before you get. So this is level, it's called fertilization. Okay, fertilization. So you take one gamete, all right, you join them up. You take the female. Okay, of course, you join here lah. This is how you're going to get the next generation. This gamete, let's say let me label as number 1. This is number 2. This is number 3, and this is number 4. You need to take one from the male and one from the female in order to get your next generation, correct? So you take 1 and 3. You can't take 1 and 2 because they are both male, okay? One male gamete, you cannot fertilize. You cannot have fertilization. So take 1 and 3. When you have combined your 1 and 3, now look at the combination of allele that's inside this individual. You're going to get capital T and small t, correct? You just add them together. And this is going to be your F1. This is your anak. This is your offspring. This is the F1 generation. Okay, now this is one of the possible combination. Now there could be also other combinations. Maybe not this gamete fertilize this one. There could be another possibility. Another possibility is your 1 can also combine with your 4, isn't it? You can also have another possibility, which is your T. Also the same thing you get. All right, now so your number 1 has used up. You already used 1, 3, 1, 4. Now another possible combination is, what about this one? Your gamete 2 here, all right? It can also combine, in order to get the next generation, is to combine with a female gamete. So you're going to get another instance of big T and small t. And there's another combination which can also happen. This one 2 can also combine with 4. You will have this, isn't it? Don't you get it? All right. So now this is the possible combination of alleles for your F1. And he noticed that all of them have dominant, right? All of them are dominant. So this shows that all of them are tall. Okay, which correlates to what he already actually found it. He found the F1 all tall, no short. Okay, all tall and no short. So now let's carry on to the next generation. Remember, he repeated for the next generation, F2. So what he did was he took, all right, F1, he self-pollinate. Okay, so this is F1. All right, this is F1. So now he self-pollinate. So he's going to have the next generation. So this is going to be F1 generation. He self-pollinate, then he's going to, of course, you need to produce your gamete first before you have the next generation, isn't it? So again, now this F1 will be producing this type of gametes. Okay? You see now? They are not pure anymore. The F1, most importantly, you can see they are not pure. They are not pure breeds. They are not pure breeds. They have a combination of dominant and recessive. Okay, all right. Okay, nothing else so far. So let's carry on. Now, so you have to write back here. Don't forget, write meiosis here at the side. Meiosis, you must write. Sorry, this is not meiosis. Yes, correct. This is meiosis. The next level is gamete. Yes, this is a gamete. Now, the next thing you need to do is you make sure you got to combine your gametes in order to get your F2. So again, repeat using the same formula which I've given you. Make sure you use up all the possible combinations. Okay, what are the possible things you can have? This gamete and that gamete. So you're going to get capital T, capital T. Now, complete one. Don't draw all the lines and then you fill up. You're going to get confused. You're definitely going to make mistakes. So you combine the 1 and 3 and fill up your F2 first. This is F2. This is fertilization. Okay? Okay, just draw and write as I go along. Okay, the next combination will be 1, 4. Yes, you're going to get another possibility. And this one is not the same. You're going to have a dominant and a recessive. You can see that? All right. And then what about the next one? This one can also combine with this. You're going to get capital T, small t. Usually we write the capital first. All right. You can also write like this, but this is less common now. We normally write the dominant first. Doesn't matter. You can change the position. You can write your dominant first, recessive first. It still has the same thing. Okay. It's still referring to the same thing. Then you can have this and this. You will have small t, small t. Again, this is different already. Different from up here. Your F2 will have different combination, right? So now what you need to do is you write down the phenotype. Phenotype means what is the characteristic. When you write phenotype, you write in words, whether it's tall or short. When you write genotype, now this is what I mean by genotype. Genotype, you write the capital letter and the small letter. That's called genotype. Genotype tells you specifically what kind of genes or alleles are inside there. But phenotype just tells you what is the observable. Okay, if you have capital T, what does that mean? Is it tall or is it short? So this one is tall. This is because you have just one dominant is enough to show. You don't need to have 2. So this is also tall. The recessive will not show. It will not manifest itself because it's not strong enough. Once you have a dominant, it will show the dominant already. So this is tall. And look at this one. Now this is a special one. You see, this one you don't have any dominant at all. You don't have any dominant allele, it will definitely show the recessive allele, which is short. So you can see you have a ratio of 3 to 1. 3 tall, 1 short. So this is how he explained. Every single time he did his experiment, he will always get one feature which is more, which is the, for example, here is the tall. So he said that this one must be tall, must be dominant feature, dominant allele. That's why he worked backwards, and then he got his Mendel first law. Okay, so this 3-to-1 shot, this is what we call phenotypic ratio. Phenotypic ratio. You're telling, you're summarizing that in this F2, about 75% will be tall and 25% will be short. So indeed, he did many times and many rounds of investigation, and he got this. I'll show his result. Okay, sorry, I'll show his result. Here, you can see he did height, he did on height, he did on flower color, flower position, all the 7 types, and he crossbreed 2 contrasting traits. Sorry, and he always found an F1. He only showed 1, and this happens with the dominant feature. This happens with the dominant trait. We call it dominant trait. Okay, in F1, it all shows the dominant because they are already become like this. Is capital T, small t. So one dominant allele is enough to show the feature. The other recessive is already masked, they covered already. Okay, so and then in F2, you will always get, you can see the one that is dominant trait will always be more. You see the ratio? It doesn't matter how many investigation, how many anak or offspring he get in the second generation. Let's say 7, 8, 7, and 2, 2, 7, 7. Roughly the ratio, roughly, if you count, you do, of course, it won't be exactly the same 3-to-1. It's roughly, you can see all this data here. It's always roughly 3-to-1, which is the dominant trait will always be 3 times more compared to the recessive trait. So this is, he said, this one must be a law, because when I'm doing it hundreds and thousands of times, I always get the same result. That means there must be a law to it. Okay, and then he found this law. He said that this must be true. This must be true. A law means it's always true. It's a universal law. Okay, that a law is something that is, like, for example, the sun always rises in the east. This is the law. Okay, there's no such thing sun rising from the west. So it's definitely a law. So, and then he found that in order to explain all these data, this must have happened. This must have happened. Otherwise, you cannot come to 3-to-1. Okay, you cannot come to this ratio of 3-to-1. So that is what he did, right? That's why we call it, we credit him with the father of modern genetics. Because without what he started, we wouldn't have known so much about inheritance. Okay, he started with this first law. So let's go through. I hope you understand this. Now, this is nothing much. It's just to explain what I've already explained just now. Okay, formation of gametes, you must separate. Okay, let's go through very briefly. One gamete can only carry 1. And then in the F1 generation, they combine together, and then you're going to get your TT, right? A capital T and a small t. And then for the next generation, right, you're going to have a combination. You have 2 capital, this is a capital T, this is a small t, and it's a capital T and a small t for the F2 just now. That's why you're going to get capital T here, capital T, small t, capital T, small t, and this is a small t, small t. So you always get 3, this is 3, this is 1. Okay, now, so let me see now what else I've got to explain to you. Now, what I want you to do is you apply your Mendel's first law and how we do this crossing to various questions. So I need you to master this first. You need to master how to draw this. You first of all, you always start by getting your key. Okay, remember, because we're going to give you marks for it. Let's say this whole diagram carries 7 marks. If you forget to write your key, you will lose 1 mark. Okay, and every level, when you write this one, we may give you 1 mark. Okay, let's say here, your pure bit is correct. Okay, 1 mark. Your gamete, the combination, another mark. You see where your marks come from. This one are everything correct. If any one of them wrong, you are going to get it wrong. Okay, so the next one, this one is correct. So that's why you get your marks all coming in. Okay, everything must be correct. And let's say they ask you the phenotype, everything. So you can see you can get 7 big marks just for 1 diagram. But if you miss out drawing the left-hand side, which is here, you didn't write here. All right, I've heard before, people from mark SPM before. This is their scheme. All right, if you do not write this one, you don't get this. That means this one, you don't get it. Habis lah. You already get only 3 out of 4 or 3 out of 7. You can understand, right? Okay, you don't get this if you do not have this. Okay, one more thing. I want to show you another way of writing it. It's called using a Punnett Square. Okay, so this one I'm not going to go through. This is essentially what I've mentioned now. You just fill up the blanks with the correct symbols. All right, just what I did here was your capital T. I just go it quickly. Capital T, small t, and then after that you're going to combine. And, oh, sorry, this is capital T and small t, capital T, small t, capital T, small t. Yeah, okay. So you just fill up everything. Now, this is a Punnett Square. This is the last thing I want to explain to you, Punnett Square. Punnett Square is the same way, is the same method where you use it to show the fertilization process only. It's to show the combination is possible. It's to show random fertilization. That means this gamete can have a chance to combine with this gamete. So basically, I follow this method 1, 2, 3, 4. We just say 1, 3, 1, 4, 2, 3, 2, 4. You will never go wrong. Why I always ask you to say 1, 3, 1, 4, 2, 3, 2, 4. What happens if you do it like this? I've got students who have done it before like this. It looks beautiful. For example, let me show you. I've got 4 gametes here. I want to combine them. Okay, so this is 1, 2, 3, 4. That's a capital T, small t, small t. All right. And what she did was, did like this. Simply draw the line. Simply draw the line like this. Okay, like this. I don't know how she did that, but it was very beautiful. Okay, it was very beautiful, and she did it like this, for example. Hey, sorry, not like this. Let me see. I don't know. I think she did it like this. That's why it looks so nice. Beautiful. This. Okay. Wow. Symmetric, right? Symmetrical. One. Let's see what the combination is. That correct or not? Is that correct? Let's see. First one, you get a combination from what? 1 and 3. Okay, 1 and 3. So you got 1 and 3. You have already used 1 and 3 combination. Now, what about the second one? This one, the next one, this one. Also 1 and 3. What happened here? Here she repeated. You shouldn't repeat because you're talking about the possibilities. Possibilities, if it happens that you have one possibility, then you must take another possible occurrence. You must try for another one. So that you cannot be repeating 1 and 3, 1 and 3. So this method is definitely wrong. It looks so nice, it looks so wonderful, okay, it looks so beautiful, but this is not the correct way. Make sure you follow my rule here. 1, 3, 1, 4. The next one you should try is 1, 4. And then 2, 3, and 2, 4. You get what I mean? Okay. So you don't draw like this. This one again is going to be 2, 4. She put a 2, 4 combination, and then you find here it's repeated 2, 4. So this is definitely wrong. So if I see something so nicely symmetrical, I know this is wrong already. Okay. So it has to be like this. Make it a practice how to draw it. It's very simple. You just draw 1, 3 and fill up first. Do not draw all the line and then you fill up. You're going to end up doing the wrong thing because all TTT the same. You're going to fill up the wrong thing. So 1, 3, 1, 4. 2, 3, 2, 4. And you will never go wrong. Okay, so back to the Punnett Square. Punnett Square is a different, just another way to show the combination. Okay, so here what you do is you write all the genes here, all the possible genes, like here, gametes. Gamete, not gene, gametes here. Here. Capital T, small t, capital T, small t. Oh, sorry. Here. This, not this. Big T, small t, right? You can see here, big T, small t. You put it here. You draw a square. You see, big T is from one individual. The small t, another individual. So you just combine them and you put this one together. From the top, from the left, you join up, you get capital T. So here in F1, they are all tall. Okay, big T, small t, all combined together, you get this one. Now for the F2 generation, F2 is this one. F2 now you have capital T, small t. Okay, you can see here, capital T, small t here, one individual. The other individual will also have capital T, small t. So you put it this way. One individual, you put like this. Capital small t. I rub this away first. And the other individual, you put like this, capital T, small t. So you start combining them. So the first one is capital T, right? Capital T, you combine with what's on the left-hand side here, you get capital T, capital T. Then the next one, you combine capital T and the small t, you get this. The next one is you get this. The next one, you get this. You see? This is the next one, different. So this one, you will get 3 tall plants and 1 dwarf plant. So Punnett Square is just simply, it's a method to represent all these lines. Oh, sorry, what happened? So sorry. Okay, sorry. It's just to represent all these lines only. Okay, sorry, it just flew away. Here, it's just to represent these lines. This is represented by Punnett Square. So for monohybrid, no need to write Punnett Square, no need to draw, because it's simple to draw 1, 3, 1, 4, 2, 3, 2, 4. But you will need your Punnett Square for your dihybrid inheritance. Okay, next week's lesson, where you look at the inheritance of 2 characteristics at the same time. Okay, so I hope that today's lesson is clear. All right, so here, this one I just mentioned, no intermediate. That means you don't have a medium height. There's only either tall or short. Okay, so because of this one, then he was so intrigued. How come you don't have a blending? There's no blending. There's no blending because the alleles are very discrete. It's either this or that. Okay, there's no blending. There's no medium height. There's either tall or there's either short. Okay, so this is what I wanted to know. So you make sure you do a lot of practice doing this, drawing this. Otherwise, if you master this, you'll be very happy when questions come out, because you're confident enough you can answer all the questions related to this. Okay, so you have to master this. Okay, master this by doing a lot of questions. All right, so okay, I think I've gone through everything I need. Okay, you may take a screenshot of this. I want you to carry over the concept monohybrid inheritance to solve this problem. Okay, afterwards we'll discuss this in our class in Google Meet, so don't run away. So for the other students who are joining my class today, I will show you the answer in my next lesson before I start dihybrid inheritance. Okay, so for those students who want to do this, please do try this question. All right, now here in humans, we can apply the same concept because the Mendel's law also applies. It's a universal law. It applies to animals, it applies to plants. So the ability to roll the tongue. Okay, let's say now I'm rolling my tongue. You can see, there are some people who cannot do it. So they have called it the inability to roll tongue. So ability means you can do it and you cannot learn it. It is actually you're born with it because it is determined by your gene or the allele. Okay, so the ability to roll the tongue, they have found that it's dominant over the inability to roll the tongue. So there are some people who can because they have the dominant allele for rolling the tongue. They can do it. Okay, and there's some people who cannot, so they will have the recessive allele for it. So here, what happens if, let's say, the father, I put left hand side as a father, the father is able to roll the tongue, right? And the mother is unable to roll the tongue. See what happens to the children. How many % will be able to roll? How many will be unable to roll? Okay, unable to roll, I put B now. Okay, let's say. See, father can roll, mother cannot roll. But look at the combination. This is a big B, dominant, dominant. This is recessive, recessive. Okay, see, you complete the diagram and then see how many % of the children can roll, how many cannot roll. Okay, next case number 2. These are 3 different cases. Case number 2, question number 2, father can roll, but you see what's the difference here? Father can roll, ability to roll, but you see it is big B, small b. Okay, you have a different case. Actually, this is heterozygous. Just now the father was homozygous dominant. Now the father is heterozygous, and the mother also can roll the tongue. The mother also can roll, but she is also heterozygous. See how many % of the children can roll, how many cannot roll. Okay, this is interesting. You please try it. The next one. What about the second one? The third one. Here, able to roll tongue. Father able to roll the tongue. All right, but he is heterozygous. And now the mother is unable to roll the tongue. And she has to be, of course, heterozygous, homozygous recessive. Different case. You see, this is a different case. So you have 3 different cases. And again, find out how many % of the children can roll, how many can roll. Okay, so for those out there, I will show you this answer in my next lesson before I start dihybrid inheritance. Okay, so we will say bye-bye now. All right, okay, it's already 1 hour. Okay, I didn't know actually take so long because I really, really want to explain. I really want to make sure that you understand. Okay, so I take the trouble to really explain. Okay, so I will see you, my 5S2, in the Google Meet. So for those students out there, all right, so do drop me a line if you have any questions. Okay, I'll be most happy to have to answer them. So, and hope you will carry on and follow my next lesson. I'll give you the answer to this. Okay, bye-bye now. Okay, bye-bye. I'll see you in my next lesson. Bye-bye.